Welcome back. Today we're going to be doing the blood lab virtually. So, this lab examines red blood cell indices for humans using some samples from McGill University in Canada and a volunteer's blood. What are indices? Indices are calculations that provide information on the physical characteristics of red blood cells. What are they used for? They're used to determine if someone suffers from various types of anemia. In order to do these calculations, you need to first determine a person's hemoglobin concentration, hematocrit, and red blood cell count. Notice the units of measurement. So hemoglobin concentration, grams per deciliter, right? Hematocrit is percent packed red blood cells, and red blood cell count is in millions per cubic millimeter. So red blood cell count on average is 5.0 million per cubic millimeter. So you're going to figure these out for the volunteer. And then once you've determined these indices, you're going to use them to calculate the mean corpuscular volume for the volunteer, mean corpuscular hemoglobin, and mean corpuscular hemoglobin concentration. You should know how to do these calculations for the lab exam. I will give you your hemoglobin, hematocrit, and red blood cell count, and I'll give you the equations. You should be able to plug them in and get an answer, and then tell me if that answer is normal or not. So first off, read the lab handout. I know the lab handout is long. The two crazy labs would be the molecular activity and then the blood lab. And so you're on the last one of the two. So congratulations. You're going to follow the instructions for the lab. Okay, the data are on the following slides. If you can't see the videos, I'll post a link to the videos and the modules as well. Make sure your computer is updated though. So measure the values on the following slides, put them in your lab handout, and then do the mean calculations. I want you to check over your values with your lab partners. I don't want you to do I don't want you to take their calculations as word. I want you to do your own calculations and your own measuring and then compare answers, okay? It's good to check with your lab mates, especially since we're virtually. We're going to use a virtual physiology blood lab from McGill University. So there's a red blood cell indices lab. They have a lot of excuse me, they have a lot of virtual physiology labs. One of them is a blood lab. There's a white blood cell differential count. I think there's an EKG and what have you. So this is a really good resource, you guys. So make sure that you spend some time looking at McGill University's virtual physiology lab. Okay, so we're going to use that for our blood count because it was too hard for me to do the blood count in the lab when I went in the other day. The lab does show all the blood tests. You can use it to study. Here's a website for McGill University. Okay, so the first thing is blood typing. I covered this briefly in lecture because I really want to explain it in lab. All right, so first off, does anyone know their blood type? Okay, so what's your blood type? All right, so what exactly does that mean? So for example, my blood type is A positive. What does that mean? That means that I have an A on the surface of my red blood cells and I have an RH factor on the surface of my red blood cells. How do I get those codes? How do I get those antigens on the surface of my red blood cells? I'm born with them, but I have a genetic code, right? So I have um, a chromosome, and on that chromosome, I have a location or a gene that codes for blood typing, okay? So blood type, so when you think of somebody's blood type, for me, my blood type is A positive, okay? What does that mean? That is telling me what antigen is on the surface of my red blood cell. So here's my red blood cell, right? And on the surface of my red blood cells, I have A's. What does the plus code for? It actually codes for the RH factor, okay? How do I get these? I'm born with them, first off, right? But we call this a genotype. So the genotype is the two allele or two trait combo that codes for blood type or two gene. It's not really a gene. Allele is a A chromosome has many genes on it, and there's a gene that codes for blood type. An allele is one possible um, combination for that, or one possible blood type for that, and then you have another one. So let me explain this. I'm not explaining it very well. I'm going to draw chromosome number 9. I know my pictures are beautiful, right? That's chromosome number 9. And so um, let's say blood type is coded for right here. Okay, so this gene codes for blood type. Okay. Um, this is chromosome number 9, but this is one of the pair. So this is called a chromatid, right? Sorry, TID. Sorry, guys. And this is a chromatid, right? Where did I get this chromatid from? I got this one from mom, right? So that came from my mom. And then where did I get this one? Good, it came from my dad. Okay, we talked about this a little bit in lecture. So this is chromosome 9 or chromosome pair 9. This is the location, or this is the gene that codes for whatever blood type I am. And let's say for here at this gene, I have a code for A. Okay, so this allele of the gene codes for A, or this, I can't think of the word that you would, like a normal word, this one of the codes for A. And then I got the other one, I got another code from dad and he gave me an A. So I have an A code from mom and an A code from dad. And so that's going to give me A on the surface of my red blood cells. Okay, and if I have A on the surface of my red blood cells, that's going to dictate my blood type. So when I say I'm A positive, that means I have A on the surface of the red blood cells. Where is a positive coded for? I think it's coded on chromosome 9 too, but we'll just say it is, and we'll say it's right here. Okay, so in order to be positive, I have to have, that means I'm Rh positive, right? I have the Rh factor on my red blood cell. And so the Rh factor code, whoops, I overshot. Let's say this would be a code for the Rh factor. And then this would be a code for the recessive factor. But since I have one dominant code and one recessive code, I'm gonna display the dominant code, okay? Let me explain dominant. So if a trait is dominant or recessive. So um, let's back up a second and say a trait that's dominant is going to trump a recessive trait. Okay, so let's go over a couple of examples. So hair color. If you were to look around the lab room, right, what hair color would be dominant, dark or light? Dark, okay? So if you have a code for dark from on one chromosome, and a code for dark on another chromosome, what hair color are you going to have? Dark hair, right? If you have a code for dark on one chromosome and a code for light on the other chromosome, what hair color are you going to have? You're going to have dark hair color, right? Why? Because dark hair color trumps light hair color. The same holds for eye color. So which eye color is dominant? Light or dark? Okay, dark eye color. So you could have two dark alleles of that gene to display dark eye color, or you could have a dark eye color and a light eye color. And because dark is dominant, you're going to have dark eyes or brown eyes, right? What gene or what allele do you have to have what allele do you have to have light eye colors? You would have to have two recessive genes, okay? So you'd have to have a recessive gene for light, and you'd have to have a recessive gene for light. That's the only way you can have light eyes, okay? Because if you have a code for dark, you're gonna display dark eyes. Okay, I apologize if I lost you. Just note too that eye color is coded for by 3 genes, not just one, so I made it way simpler. Okay, so so we have different blood types. We have blood type A, and I'll go over it in a little bit more detail in a minute. We have blood type B, we have blood type A, B, and O. So those are our choices for ABO. A and B, you guys are dominant over O. So these guys are dominant, and O is recessive. A and B share dominance. So we call that an example of codominance. Okay, so if you're trying to look up examples of blood type, you can look up codominance as well. I think the Khan Academy has a good one for MCAT review for blood typing. Okay, so A and B are dominant over O. O is recessive. Positive for the Rh factor. So if we look at the Rh factor, positive or having the Rh factor is dominant over not having the Rh factor. Okay, so this follows straight genetics. This is dominant and this is recessive. Okay, let's actually go through the PowerPoint, right? So blood type A. We have blood type B, right? Blood type AB. And what's the recessive one? Oh, good. And then for Rh factor, so this would be your AB, whoops, this would be your ABO blood types, right? Let me actually get the laser pointer. This would be your ABO blood types, right? A and B are dominant over O. So these are codominant over O. Then what about your Rh factor? So we can be positive, right? That denotes that we're Rh positive, that we have the Rh factor. Or we could be negative. That denotes that we're Rh negative, that we don't have the Rh factor on the surface of our red blood cells. All right, let's go through the genotype or the two allele combination on chromosome 9. If we have blood type A, what two alleles could we have on chromosome 9? So we could have 1A from mom and 1A from dad, or we could have an A from mom and an O from dad, right? If we have two A's, that's going to give us blood type A. If we have an A and an O, remember A is dominant over O and O is recessive, so we're going to display A on the surface of our red blood cells. Okay, what do you think for B? So fill it in. Okay, so we could have a big B from mom, a big B from dad, or a big B from mom and an O from dad. We could say an O from mom and a B from dad as well. I just didn't want to take way more time. Not that this already doesn't take time, but excessive time. Blood type AB, you have to have an A from one parent and a B from another parent, okay? Because you have both of them and neither one is dominant over the other one. They're sharing dominance. You're going to display both of them on the surface of your red blood cells. What about O? What does O code for? O is recessive. It codes for the absence of A or B, so it codes for nothing. So if you have blood type O, you have to have two recessive alleles, right? You have to have an O and an O. I usually do Rh after I do these, but since I didn't design the PowerPoint that way, we're going to cover Rh. So if I'm Rh positive, what two alleles could I have? I could have a positive and a positive, right? Or a positive and a negative. Positive is dominant, so if we have a positive and a negative, the positive trumps the negative. It's just like cards, right? And we're going to display the Rh factor. What two alleles do we have to be Rh negative? We have to have two negative alleles, right? We have to have two of those recessive alleles. Good. So since we have these genes or these codes on our chromosomes, these are actually going to code for the antigens on the surface of the red blood cell. So let me draw you a picture. This is so weird, you guys doing it on a PowerPoint compared to in a classroom on a whiteboard. Okay, so here's my red blood cell, right? Remember, we talked about glycolipids or glycoproteins, and those are antigens, are markers on the surface of the cell to identify our cells as us. Well, these are antigens. These guys code for antigens on the surface of the red blood cell. Why do we care about these guys? Because these guys are going to cause an immune response when we talk about donor and recipients. Donor donating blood, recipient getting the blood. So antigen. You can't talk and write at the same time, okay? So antigens are on the surface of the red blood cells. So if I have blood type A, I have the code for A. What am I going to display on the red blood cell? Good, I'm going to display A on the surface of the red blood cell. I have the code for B, so what am I going to display on the surface of that red blood cell? B. Awesome. I have the code for A and B, so what am I going to display on that red blood cell? I'm going to display both A and B antigens. They're not stuck together, we just have both the codes, so we're going to display both of them. Blood type O, what does O code for? O codes for nothing. So the surface of that cell is going to be smooth. There's not going to be any antigens because O codes for no antigens. Okay. What if we're Rh positive? We have the code for Rh, so we're going to display Rh on the surface of the red blood cells. What if we're negative? Negative codes for no Rh, right? So the absence of Rh. Okay, so this is where you need to pay attention. So far, it's pretty straightforward. Our genes tell us what's on the surface of our red blood cells or what antigens are on the surface of our red blood cells. And from there, that gives us whatever blood type we tell everybody, right? I want to back up though. So whatever antigens we have on the surface, we are going to make antibodies against foreign blood. Okay, so these are the antigens on the surface of our red blood cells, right? We have the genetic code for those. We're going to make antibodies against foreign blood. So antibodies are gamma globulins. They're in the plasma. They're found in the plasma. If I have A blood, what antibody am I going to make? So if I have A on the surface of my red blood cells, I'm going to make antibodies against foreign blood. What is that foreign blood, guys? Good. So I'm going to make B antibodies, right? Let me go back and draw my picture. So here's my red blood cell, right? It has A antigens on the surface of it. That was from my chromosomes. Okay, that was inherited from mom and dad. Because I have A on the surface of my red blood cell, my body is going to make B antibodies. Remember the antibody in lecture? It's that sideways Y-shaped. And so I'm going to make antibodies to attack foreign blood. And foreign blood is the opposite blood type. Okay, how do antibodies work? Let me cover that real quick. Let's say I have an antibody. Okay, let's say I have coronavirus, right? So there's my coronavirus. And here's my other coronavirus, right? How does that antibody work? My immune system, when I get a virus or bacteria or foreign entity into my body, my immune system is going to make antibodies against it, right? So when we have foreign invaders, one mechanism we have to attack them is antibodies. So how does that actually work? That antibody, let me change the pen color so you can see it. That antibody is going to bind the coronavirus. Whoops, sorry. So here's my sideways Y, right? There's my antibody. And this antibody is specific for coronavirus, right? It's not going to bind bacterial pneumonia. It's not going to bind spinal meningitis, right? It's specific for corona, COVID-19 more specifically. So that specific species of virus. So usually when we have a viral or bacterial infection, we're going to make antibodies against them. The antibodies are going to agglutinate them, right? Agglutinate, A-G-G-L-U-T, nate, which means stick together, so that your white blood cells can come in and phagocytize them, okay? So if it's a viral infection, it's going to be lymphocytes and most likely, lymphocytes and monocytes, okay? And it depends. Sometimes they're slightly different, but that's what happens. Okay. And so once you have this antibody, you have it. Usually, measles actually wipes out your immune memory cells, but once you have this antibody against COVID-19, you have it. There's still literature, they're still trying to figure out everything, and I don't want to digress and take up all of your time, but coronaviruses, I guess we have a weak immune response to them. And so we have a low level of antibodies being produced when we're exposed to them. And so it's easy to get an infection when we are exposed to them again. So really interesting. We're still trying to, we're still, they're still doing research on this. Okay, so anyway, whatever is on the surface, whatever antigen I have on the surface of my red blood cell, I'm going to make antibodies against foreign blood types so I can attack those foreign blood types. Okay, so if I have blood type B, what is my antibody going to be? What's foreign blood? Good, A. So I'm going to make antibody A, right? If I have both A and B on the surface of my red blood cells, am I going to have an antibody? No, because I would attack my own red blood cells. You're not going to have an A because you would attack your A's. I'm not going to have a B because I would attack my B on the surface, my B antigen. It's important that you guys get practice using antigens and antibodies and you don't mix them up. If I have nothing on the surface of my red blood cells, what antibodies am I going to make? Good. I'm going to make both antibodies, right? Because I don't have anything on the surface of my red blood cell. And so I'm going to make antibodies against foreign blood, and that would be A and B. What about Rh? So if I have Rh on the surface of my red blood cell, am I going to make an Rh antibody? Why not? Because I would attack myself, right? So we don't make any. What if I am Rh negative? I don't have anything on the surface of my red blood cell, and so I'm going to make the Rh antibody, okay? There's a trick to this one though. You don't make this one until you're exposed to Rh positive blood, okay? So this behaves similar to what happens when you come in contact with a virus or a bacteria, right? These antibodies in the plasma, so your A and B antibodies, you are born with those, okay? You come out of the womb with those, so you're born with. This one, you don't make until you're exposed to foreign blood. So you don't make the Rh antibody until you're exposed to Rh positive blood. And so that follows the same line as, oh, I came in contact with bacterial pneumonia. I'm going to make an antibody against that specific species of bacteria, right? Oh, I have COVID-19. I'm going to make an antibody specific for COVID-19. Immunizations are important because they are dead or weakened versions of the virus or bacteria, and that allows your immune system to make antibodies to attack it. And once the antibodies attack the weakened version or the dead version, that will allow your body to remember those, the virus or the bacteria, and you'll be able to get an immune response really quick or antibodies produced really quick the second time you encounter it. So the first time you encounter a foreign bacteria or virus, it takes about 14 days to have a full immune response. That includes, making your antibodies and your whole cavalry, your white blood cells coming in, et cetera, et cetera. The second time you come in contact with a foreign antigen, right, a bacterial viral antigen, you're going to have a full antibody and immune response within 48 hours. So it happens really fast. So that's why vaccines are recommended, because the second time you come in contact with that virus or bacteria, you're not, your body's going to be able to handle it and not be overcome by it. Okay, I filled in the table for you. There you go. Okay, so can you apply that, right? So what I recommend is drawing out this table, 5 to 10 times. Make sure you understand it. So that's the big part. I can help you. Tutoring can help you. You can explain it to yourself. Explain it to your lab mates on Zoom. Zoom actually has a whiteboard feature. So if you share screen and you click on the whiteboard, you can actually draw it out and explain it to each other. So that's a really good. My cat just walked over my computer. That's a really good way to do it. Okay, so can you critically think about this knowledge, right? So can an A positive person donate red blood cells to a B positive person? I want you to draw it out and explain, right? Really important. Okay, so how do we figure this out? And you can make up more of these. I just have 2 of them that I'll go through with you. So go ahead and pause this, work it out, and see if you come up with the right answer. Well, the same answer that I come up with. How are you going to attack this? So I want you to draw the antigens and antibodies for this person. And notice that I'm just talking about red blood cells, right? So when you're donating blood, it's going to be either packed red blood cells or it's going to be plasma, because you want the platelets and, the albumins and whatnot. Oh, antibodies, right? And to a B positive person, okay? So pause it, figure it out, and then start it back up again. Okay, so A positive, what does the surface of their red blood cell look like? Good, so they have A's on the surface, they have A antigens, and what does a positive mean? Good, they have the Rh factor, okay? So now you know what's on the surface of the red blood cell. What is in the plasma, right? Here's your red blood cell. The sideways Y denotes that it's an antibody. So a quick trick is whatever's not on the surface of the red blood cell, that's some antibody that you're going to have. So I don't have the choices are A, right? Your choices are A, B, and Rh. Those are your choices. So what don't I have? B. So I'm going to make B antibodies, right? So A positive, I have an A, I have an Rh. I have a B antibody. This is going to attack B blood, okay? What does a B positive person look like? Here's my red blood cell, right? They have B antigens. What does that positive mean? Good, they have Rh antigens as well. What antibodies do they have in the blood? And it doesn't matter what direction you draw these, you can draw them in whatever direction. Okay, so they don't have A on the surface, so they're gonna make A antibodies. Okay, now my trick is you have to see if I'm talking about red blood cells, plasma, or whole blood. Typically in a real world scenario, they're not gonna give whole blood, they're gonna give packed red blood cells with saline or plasma with the antibodies in it, okay? So we're going to take this red blood cell and we're going to stick it into this person. Can we put that red blood cell into this person? No. Why not? Oh, because this antibody is going to bind to this antigen and agglutinate it. Okay? We don't have any Rh antibodies, so we don't have to worry about that. We don't have to worry about B antibodies coming in contact with B antigens because we're not donating this, right? Just these red blood cells. Okay, try the next one. Okay, so that's the answer. Let's go to the next one. Can an A positive person donate red blood cells to an A negative person? Right? I'm not going to tell you the answer for this one, but I want you to be aware that it's A positive. So what's on the surface of the red blood cells? You have an A antigen and an Rh antigen. You're sticking those red blood cells into somebody who's A negative. All right, you need to think about A negative and if they have an Rh antibody. And if they don't have an Rh antibody, when do they make that Rh antibody? Solve that one. Ask your group members if you're stuck. If you're still stuck, ask the tutors or ask myself. Okay? Good. All right, so blood typing. How do you determine what blood type someone is? I think I talked about this in lecture, but I just wanted to reiterate it. You want to know what antigens are on the surface of that person's red blood cells. And so you're going to put the blood into 3 wells. So here's well labeled A. Here's a well labeled B. I know it's kind of hard to see. Here's the well labeled RH. Okay. And you don't know what blood type this is, but you want to figure out what blood type it is. Okay, so you're going to add antibody serum from that we got from like Carolina or from Sigma Corporation and see which one agglutinates. So if you take a look at the picture, oh, this should be in order and it's not. So here's antibody A serum. What does this contain? This contains A antibodies. That's why it's called anti-A. That stands for A antibodies. This is simulated, so we can't use real blood in lab. So I used the fake blood. So here's the simulated A antibody serum or anti-A serum for short. We would stick some of this antibody A serum in the A well. We would stick some of the antibody B serum in the B well, and we would stick some of the antibody RH serum in the RH well. And then we would see which one agglutinates. Whatever one agglutinates, that means that we have that antigen on the red blood cell. Okay, so let me say that one more time. It's going to take a little bit for you guys to understand. All right, okay, so let's say we don't know the person's blood type, right? So here's our red blood cell, and here's our antibodies. If they have antibodies in the plasma, they might not. We don't know what's on the surface of the red blood cell. We don't know what antibodies they have in the plasma, okay? We can't figure this out easily. Well, we could technically do a blood test, but if we're doing this really quickly, a sure way to do it is use antibodies, okay? So we're gonna stick the red blood cells or the blood into these wells, right? And then we're gonna add antibody serum. So we're gonna add A to this well, antibody A to this well. We're gonna add antibody RH to this well, and antibody B to this well, okay? And if these red blood cells stick together or agglutinate, that tells us, because we added A antibody, that we have A antigens on the surface of the red blood cells, okay? If these guys agglutinate, right, these are my red blood cells agglutinating together, that tells us that what's on the surface of the red blood cell. Good, your RH factor. And if we get agglutination in B, right, it's actually like stringiness because it's fake blood, that tells us that we have B antigens on the surface of our red blood cells, okay? So, and this works with real blood as well, right? We're not going to stick all the antibodies into one well because when we get agglutination, we wouldn't know what antibody caused the agglutination, and so we wouldn't know what's on the surface of the red blood cell. The whole point of blood typing is to figure out what is on the surface of the red blood cell, right? Once you know what's on the surface of the red blood cell, you can then assume that you know what antibodies in the plasma, right? So if I have B antigen, sorry, it's my cat again, B antigens and RH antigens, because that's what agglutinated when we added those antibodies, the synthetic antibodies or the antibodies we ordered. Then we can assume that we're going to have A antibodies in that person's plasma, right? So these antibodies are not what's in a person's plasma. These antibodies are strictly for helping us figure out what antigens we have on the surface of our red blood cells. And then from that, we can backtrack to figure out what antibodies we have in our plasma. Okay, so you're going to go through this lab and watch the video. You're going to figure out what blood types we have. So write them in your lab handout. So here's Miss Brown, Mr. Green. See this stringiness? This means agglutination for the fake blood. It doesn't look like real agglutination. Mr. Jones, Mr. Smith, and then for hemoglobin concentration is the amount of hemoglobin in 100 mils of blood. So amount of hemoglobin expressed in grams per 100 mils of blood. Normal range, 14.1 to 15.6 grams per deciliter. Keep in mind 1 deciliter is 100 mils. You should know your conversions. 14.1 is normal for female, 15.6 is normal for male. Why are male red blood cell indices higher than female? What hormone? Good. There's a question on your lab manual, or lab handout, excuse me. To do the hemoglobin concentration, we're going to use a Talquest scale, okay? So how does it work? You have a piece of chemically treated paper, you put a drop of blood on there, weight, the instructions are actually right here, right? Wait about 30 seconds, and then you're going to match up the color on the strip with the color of the scale. So this is the color scale. Take a look at the scale. So normal 15.6 grams. Keep in mind that this is not the full measurement. It's concentration. So it's supposed to be weight over volume, or grams per 100 mils, right? Or grams per deciliter. Don't give me percent. I don't want percent. So 15.6 grams per deciliter, 14.1 grams per deciliter are normal. Suggestive anemia, 12.5 grams, 10.9 grams. Anything below 10 is going to be considered anemia. All right, so if you have a hemoglobin concentration below 10 grams per deciliter, depending on what it is going on, you may need a blood transfusion. You may need packed red blood cells, right? So I want you to figure out, here's our sample. So I want you to figure out what the concentration is for this in the next slides. I'm going to zip through it so that you can figure it out on your own, right? Whoops. The next portion is part C. That's coagulation time and bleeding time. Bleeding time is going to tell you how long it takes for a cut to stop bleeding. So the vascular spasming, okay? So let's say you get a paper cut, right? And it stops bleeding. That would be bleeding time. Let's say you hit it again on accident, right? You hit it against something and you notice that it's still bleeding. Has coagulation occurred? Blood clotting or coagulation has not occurred. And so it takes longer for that to happen because you have to activate all those clotting factors. I talked about this in lecture, so go back and review my lecture PowerPoint. For this lab, you're going to use the fake data on page 10 to do your calculations and answer the questions in your lab handout. So there's fake data in the thing for coagulation time and bleeding time. We technically don't do this in lab. Normally for lab, when we're on site, we use sheep's blood, and it has all the clotting factors removed because otherwise it would be a big glob of clotted blood and we wouldn't be able to use it. So we don't do this in lab. I just have students read it theoretically and understand it. Okay, part D is microhematocrit. So you're going to tell the hematocrit of the sample. So hematocrit is amount of packed red blood cells expressed in percent, normal range, about 38 to 55%. So I would say women are about 38 to 42. Men are about 42 to 55%. It depends. I talked about this in lecture. So remember, if you have low oxygen, your hematocrit's going to be higher because you have more red blood cells in circulation. Read the instructions on how to do it. Here's the capillary tubes. You should be able to identify that. Here's a capillary tube with blood in it. So I sucked up the blood, right? I put a clay in one end so that the blood doesn't slide out of the glass tube. It's like a glass straw. And then I put the clay side out in the centrifuge on slot 1. This is a microcentrifuge. I'm going to spin it down, and the red blood cells are going to collect towards the end, and the plasma is going to collect in this region. Red blood cells are heaviest, so they're always going to go towards the outside. It's a centrifuge. Take a look. Super cool. This is the blood sample after it was centrifuged. Here's a packed red blood cells, and then right here is a buffy coat. You can't see it very well, but it looks like this white sort of layer, and that is where the white blood cells and the platelets are. And then you see this golden color. This is actually plasma, and so it should be clear and golden. If it's clouding it in golden, it means you have too many triglycerides in your diet when you last ate. So I used to work as a plasma at a plasma center when I was in undergrad, and I used to be a technician that dealt with the plasma after the phlebotomist collected it from people. And you could always tell, because it was a college town, you could always tell who binged and went out drinking and eating a bunch of junk food, because their plasma would be really cloudy and opaque because it had so much fat and they were so dehydrated. It was really gross. And then people who ate really healthy, their plasma was this golden clear color. Okay, so just a little bit of insight. All right, then here's a hematocrit reader card. You can watch the video on this snippet. And then you're going to measure the hematocrit. I explained it in the video, so go back and look at that. The last one is a red blood cell count. So this is part 2. I actually took 2 labs and stuck them together. The red blood cell count is the amount of red blood cells in a volume of blood. Be careful, because this is expressed in millions per cubic millimeter. All right, so you don't write your answer as let's see, 3,800,000 millions per cubic millimeter. It's 3.8 million per cubic millimeter, 5.6 million per cubic millimeter. Normal range, females 3.8 to about 4.5, and then males 4.5 to 5.6, right? To do red blood cell counts, normally we would use a hemocytometer in lab. It's a cell counting chamber. If you've used a hemocytometer, you know that it's difficult to use them. It's a skill set. But so I want you to read your lab on how to use a hemocytometer. And then you're going to do the counting from the McGill University website. So there's actually, here's your hemocytometer. Hemocytometer means a cell counting chamber, like a blood cell counting chamber, okay? You can actually use it to count red blood cells or white blood cells. Here is a mirrored edge, right? A mirrored face, and here's another mirrored face. Right about here is where we have a grid for counting red blood cells and white blood cells. Here is another grid, okay? Typically, I have students find the grid first on a microscope. Then they come to me and get diluted blood. I'd stick a cover slip on it and then aliquot some diluted blood or discharge diluted blood into this grid. It's a known volume, so you can figure out how many red blood cells are in these 5 boxes. And then back calculate to figure out how much is actually in the blood of the person, right? So if you did this with your own blood, you could actually go and figure out what your red blood cell count is, okay? You're going to go to McGill Universities. It's a university in Canada, and it's actually a really great physiology university. So you're going to go to the virtual physiology lab, go to the red blood cell indices experiment, go to the red blood cell count to count your red blood cells. Here's their website. You're going to see the page that looks like this. Okay, so when I talk about the grid, this is the grid I'm talking about, just in case you're wondering. When you do a red blood cell count, you focus on this box or this grid. I think this is labeled grid 5 in your handout. And notice how I count the outer edges. So we're counting the red boxes. So I'm going to count the red blood cells in this box, this box, this box, this box, and this box of grid 5. You should have about 100 red blood cells per box, right? Note areas of the grid where red blood cells are counted. If we were counting white blood cells, if we were doing this with real blood and we were counting white blood cells, we would actually count the blue areas. So we would count this one, we would count the white blood cells in this one, we would count the white blood cells in this one, and the white blood cells in this one. So it's really interesting. If you remember, why do we have two different grids? There's way less white blood cells than red blood cells. And so you need to make the grid smaller because there's way more red blood cells. So you're going to magnify that, okay? So you're going to click there to open up a window, which actually mimics what you might see through the 40X objective. I want you to go to the four corners and count the red blood cells in each box, and then go to the middle box and count the red blood cells in this box, okay? Here are the rules. You don't have to quite follow those rules. It's not like we're getting certification for this. Here's normal ranges. So the other thing I love about McGill University's virtual physiology lab is they give you the normal ranges too. So they actually have a white blood cell lab, a differential white blood cell lab. So you can actually look at that too for a past lab that we did on white blood cells. Okay, you should have roughly 100 white blood cells per red box and 500 red blood cells total. I want you to compare your values with your lab mates just to make sure you guys are on the same page. This is actually new to me. I didn't go in and count the red blood cells, but they look close to normal. Once you have your volunteers' hemoglobin concentration, hematocrit, and red blood cell count, I then want you to use those to calculate the mean values on the back page of your lab. It might be hard to read the equations in the handout, so use the equations for these calculations in your lecture PowerPoint. All of the stuff that I've discussed in this lab is fair game, right? So you want to be thinking about what could I have you identify, what could I have you do, could I have you read hematocrit, et cetera, for the lab exam coming up. Okay, study well, you guys. This is a fun lab to do, and I'm sorry you can't do it in person, but I hope you enjoy this. Talk to you soon.